Hey guys, it's Professor Tiffany Martin and with Medical Microbiology, and we are been in chapter 15. We have been talking about our second line of defense, working through our concept map. Talked a lot about cells, and we have been in the middle of talking about chemicals. Last video you should have had talked about our TLRs and our nod proteins and how they're similar and how they're different, and how they are in the innate immune system. And then we talked about interferon and how that can protect cells, especially those infected by viruses. The last one we're going to do in this chemical section is called complement. This is probably the biggest concept in this chapter. I put you in a mastering activity also into this section so that you can work through that. And for those of you that like to do those types of things, there's it's just a good way to kind of work through it as well. So I'm going to go through PowerPoints and we'll see how it goes. Okay, so complement. So this is a chemical second line of defense. The bacteria pathogens already inside your body. What is something your body will do immediately against any foreign invader? Doesn't matter what it is, it will do this. One of these is the complement cascade pathway. So what is complement? You'll hear complement and complement proteins, complement cascade pathway. Complement are proteins. They're serum proteins, which tells you where they're located. They're in the blood, the liquid portion of the blood. They're proteins, and they react with one another in a series of reactions called a cascade. So that's why they call it the cascade, complement cascade. Basically, our body is making these proteins, cells in our body constantly making these proteins. They're constantly being secreted and in the circulatory system, and they're doing things. And what they do is there's a whole bunch of different ones. They've all got numbers and letters and stuff, and they run into each other and they bind and they combine and make a bigger protein, and then that one will break up into smaller proteins, and those smaller proteins will recombine with other proteins, and it kind of forms these series of reactions. And that is always going on. You can kind of think about it like a rock slide. Have you ever seen, hopefully you haven't seen, but think about a boulder at the top of the mountain, and that boulder starts rolling. Maybe it's only a rock. It starts rolling and it hits one rock, and that rock hits a couple of rocks, and those rocks hit together, and all kind of does all these things. That is a cascade reaction. So complement proteins are proteins we're always making. They're always secreted in the bloodstream, and they're doing things. They're forming together, breaking apart, other pieces forming together. Okay, so in comes a pathogen. So in your rock slide, you've got a big boulder sitting in the mountain, and your cascade of rocks are coming down the mountain. If there's no boulder there, the rocks continue doing what they normally do. If there is a boulder, meaning, and I'm going to go ahead to the next picture of that, meaning a pathogen is sitting in the way. For this one isn't a, do I have one more picture of this? Yeah, let's do this. So this pathogen is sitting in the way. It is in your bloodstream now. Your complement proteins, all these C proteins with numbers are complement proteins. They're being made, they're combining, they're breaking apart, they're recombining with different types of proteins. This is going on, but suddenly we have a big pathogen sitting in the way. Well, some of these complement proteins will end up binding to this pathogen, and that will start a different series of reactions. If that protein was going to combine with something else, it's no longer doing that because it's binding to this pathogen, and it can't continue doing what it's going to do, so it causes a different series of reactions to occur from that. So this is how the complement cascade gets started. If this is my pathogen, the blue oval circle here, here's my complement proteins. This complement protein binds to that pathogen and causes the other complement proteins to start doing different things. This protein is busy binding to this pathogen, and so other, whatever it was doing before, it can't do anymore. So it ends up having a different series of reactions occurring. Okay, so this is how the complement pathway gets activated. This is actually called the alternative pathway if a complement protein binds to this pathogen and changes all of these later reactions. It can actually, the complement cascade can also be activated by a couple of different ways. An antibody binding to the pathogen. An antibody binding to the pathogen. So maybe you've seen this pathogen before and you already have antibodies, and that binds to the pathogen in your bloodstream. That will also trigger a different series of reactions that we are calling this complement cascade. The third method is this lectin pathway. It's when a sugar binds this, and then you get a different different series of reactions. All right, so we have classical pathway, alternative pathway, and lectin pathway. This is how this complement cascade starts. These proteins bind to this, or an antibody or a sugar binds to this, and it starts a different series of reactions. All right, so if that pathogen is present and we get a different series of reactions, I don't need you to know what all that is. I need you to know the outcomes. So what happens? Outcomes are some of these will cause inflammation. There's inflammation right there. If this pathogen is present, these complement proteins will trigger inflammation, and we'll talk about that in a future video. As part of our innate immune system. So that's something that could happen. What else could happen? Some of these complement proteins are going to cause chemotaxis of phagocytes. All right, so you remember when we talked about phagocytosis and we saw the cell chasing the bacteria, and I said the bacteria is producing these chemical signals. That's true. Also, these complement proteins, the presence of this bacteria bound and starting this complement cascade reaction, those complement proteins can also serve as chemotactic factors. So a phagocyte can follow these complement proteins to the site of the pathogen. So that's one thing. So chemotaxis of a pathogen. The other thing it can do is cause, once it gets to that, it can act as an opsonin. If you remember opsonization, that was one of the stages with phagocytosis. It adheres to that. And we said that phagocyte is going to come and it wants to grab that pathogen. These complement proteins will bind to that pathogen, and that will help that phagocyte to grab a hold of it. Kind of serves as a handle, if you will. So some of these complement proteins will act as an opsonin. The last thing this complement cascade does is some of these proteins will bind to the pathogen in a certain series of, or a certain pattern, if you will. So some of these proteins bind, and when they bind in this pattern, it ends up creating a hole in the cell membrane. And if you end up with a whole bunch of holes in your cell membrane, what's going to happen to your pathogen? That's right, poking holes in the cell membrane means water rushes in and that pathogen is going to lyse. These series of complement proteins in this pattern is called a membrane attack complex, MAC. And so another outcome of the complement cascade is lysis. And that is because of these proteins binding and creating these holes in that. All right, so outcomes of the complement cascade. We have inflammation as an outcome. We have chemotaxis of phagocytes, and then opsonins for phagocytosis. And we have the membrane attack complex. So all of those are outcomes of this complement cascade reaction. Okay, all right, so back to here, complement activation, what's the result? Lysis, that's that membrane attack complex. They can trigger inflammation, they can also trigger fever, and then opsonization. So all of those things are outcomes of complement activation. We mentioned that it can be activated in 3 ways: classical, alternative, or the lectin pathway. Let's not talk about the lectin pathway, but let's compare the alternative pathway and the classical pathway. I'm going to give you a little quiz and let you think about it. But if we've got a pathogen and it's gotten into our circulatory system, which of these pathways will get activated first? The alternative pathway or the classical pathway? So take a second and think about that. Think about what you know about what is necessary to activate this by both of those pathways. And which one do you think may happen first when we see this? Okay, so I'll let you thought about that for a minute. The classical pathway needs an antibody. In order to have antibodies formed, we have to have seen this pathogen first. So the classical pathway, a lot of students will answer, well, the classical pathway is most likely to happen first because it's called the classical pathway. In reality, the alternative pathway doesn't require any antibodies. This will happen as part of the innate immune response. We do not have to have seen this pathogen before to activate this complement cascade. So typically the alternative pathway will happen before the classical pathway. And that's just because we don't need anything specific. This is all a non-specific binding. All right, so why do we all say classical pathway, or what a lot of students will say, tell me it's the classical pathway? Well, the name classical seems to think that it should happen first. Don't get fooled by this. This is science, and a lot of these are named by the order that they were found. So they actually found the classical pathway first, and then someone else was studying in a different lab at a different time, and they found a different way that the complement pathway, complement cascade got activated. So when they published their paper, they probably called it an alternative activation, and then maybe the first one got called classical. And so that's how these things happen. Same thing with the complement proteins. I'm not having you memorize those, but the complement proteins are named not in the order that they function, but in the order that they were identified. So don't get tripped up on that in science. So, okay, so complement cascade. You can work through that activity and see how that works for you. This is a picture of a cell here, and these little black dots are all membrane attack complexes. So those complement proteins forming a pore in the membrane, and you can see that they are everywhere. All right, this again is an innate immune response. It happens non-specifically. This happens as the result of any foreign invader. These, so you get a pathogen, you get any bacteria, gets into your bloodstream, and you start activating. We have these proteins in our blood, and they will start forming on the cell membrane. And they bind to cell membranes, and they bind to any cell membranes. I'll let you see that video in there. So if they form on any cell membrane, how come they don't form on our own cell membrane? If complement is nonspecific and just binds once they're there, and we saw how they started, the complement cascade gets started, either classical, alternative, or lectin pathways, then they're there and they bind to any cell membrane. How do we keep them from binding to our own cell membrane? Okay, so think about that for a second. What happens is in our own cells, we produce some proteins that can break down activated complement proteins. So we provide proteins that inactivate complement, and that's how we protect ourselves from this non-specific response. Okay, so watch your videos on that. Go back through it if you need to. Again, this is probably one of the more complicated topics in this chapter. Ask me questions. Thanks.